So this is our protein project. The protein chosen was myosin 2 motor domain and Dictyostelium discoidum, which is a small unicellular organism. And this type of myosin is a non-skeletal muscle myosin, which means it can be found usually if you're looking at mammals. Myosin 2 motor domain will be found in um, animals, smooth muscle like the arteries or veins, the muscle in our intestines, things like that. It's not the skeletal muscle myosin. Basically, myosin is a protein that is really interesting because it's the only reason muscles in any type of organism or any type of organism is able to move. So it's really interesting that we chose this protein, and also the way that they got the structure of the protein was X-ray diffraction with a resolution of 1.75. And we chose the motor domain, but myosin has three major domains, which are the motor, the neck, and the tail. Usually, the neck is the one that's responsible for the action of contraction. However, the motor domain is really important for contraction as well because it moves the neck so the contraction can happen. So myosin 2 motor unit binds to its microfilaments to F-actin to form contractile structures that are used for muscle contraction, cell shape change, and overall body mobility. Again, when we talk about muscle contraction here, we're talking about smooth muscle. Some organism, which is the organism in question, doesn't even have any type of muscle structure. The myosin is mostly for cell shape change and overall body mobility. The figure 2 here is showing myosin 2 motor domain, and the ribbons that are pink are the alpha helixes and the yellow ribbons are the anti-parallel beta sheets.
Hello, my name is Dat Trang, and I'm here to give you a quick overview of the structure and function of the myosin 2 Dictyostelium discoideum motor domain. We were able to determine that it has about 800 amino acids, which we displayed in figure 1 for the amino acid residues. Also, our motor domain of choice only had one chain. Typically, the myosin molecule consists of a head, tail, and a motor domain of two heavy chains. For ours, you can see it displayed in figure 2, in which you can see the alpha helices as well as the anti-parallel beta sheets of our backbone for our motor domain. As you can see, this one main chain. Myosin is a motor protein that plays a crucial role in regards to cellular processes like muscle contraction, cell division, and intracellular transport. Also, it is responsible for ATP hydrolysis. We were able to determine that there are ligands in regards to our myosin 2, and we were able to display this using JMO. The orange here is the adenosine 5 prime diphosphate, which is the ADP, so it can possibly be the area for ATP hydrolysis. In regards to myosin movement, it is a cycle of binding, power stroke generation, and detachment. And in the presence of ATP, the myosin head binds to the actin filament and undergoes conformational change, which allows it to generate the power stroke by pulling the actin filament towards the center of the sarcomere. Okay, the movement is then followed by the detachment of the myosin head from the actin filament, allowing for another cycle of binding and power stroke generation. That's the typical actin myosin transaction. All right, but there is regulation for the myosin activity. It's usually done by various signaling pathways, such as post-translational modifications. And an example is through phosphorylation of myosin by kinases.
I will be talking about the biomedical relevance of myosin 2. Myosin 2 is a protein that's responsible for generating large contractile forces that are involved in cellular activities. So disorders such as MYH9, 10, and 14 result in a progression of diseases that are due to misregulation. Lack of myosin 2 A, B, and C will cause a lot of the progression of the diseases. For an example, MYH9 that lacks myosin 2A will create a defective cell-to-cell interaction and deficiency of polarized visceral endoderm. Also, MYH14 can survive without myosin 2C, but it will require the expression of myosin 2B in the future. Continuing, we have MYH10. If it lacks myosin 2B, that will result in brain and cardiac development defects. So specifically, myosin 2A, since they release large immature platelets in the bloodstream, this causes a grand reduced amount of normal platelets. Humans are required an expression of all three functioning non-muscle myosin motor proteins to sustain development, growth, and disease resistance.